Spinning wheels that you are looking are true to the scale models of tobacco mosaic virus. This virus has a very special place in the field of virology and molecular biology. It was our first introduction into the realm of new kind of infectious agents we now know as viruses. Today's video is a story of how an agriculture problem led to the discovery of first virus. A disease in tobacco plants known as tobacco mosaic disease or TMD caused mottling of leaves and significant drop in productivity of tobacco. Farmers were worried about the situation as no obvious insect seemed to cause it. Microbiologists hypothesized that causative agent of the disease might be some microscopic life form, possibly some bacterium. Scientists of the time were under the influence of germ theory proposed by celebrated bacteriologists, which laid down criteria to link pathogen with the disease. And by invisible pathogen, scientists of the day mostly meant bacteria. So if some bacterium caused this disease, according to germ theory, one had to isolate probable pathogen from diseased organism and apply it to healthy organism. If healthy organism gets the same symptoms, organism in question causes that disease. For tobacco mosaic disease too, scientists thought that it was caused by some bacterium. So Ivanovsky in Russia collected leaves of diseased plants and extracted the sap. He used a bacterial filter to collect possible disease-causing microbe from the sap. Let's have a detailed look at the filter. Pore size of the filter was 0.45 micron, smaller than the diameter of all known bacteria by that time. Ivanovsky used this filter for his experiment. His idea was to divide sap in two portions: the residue retained on the filter and the filtrate collected in the flask. He transferred bacteria collected on the filter into a liquid medium. He had his filtrate in the flask and bacteria in the tube. He then grew two groups of plants, one that would receive filtrate and other that would receive bacteria. He sprayed two groups and waited until the emergence of disease. On day 2, plants that received filtrate started showing symptoms of the disease. To his dismay, however, plants sprayed with filter-retained bacteria did not get tobacco mosaic disease, even on waiting for many days after application. However, he still thought that it was some previously unknown bacterium, so small in size that it passed through the filter. Failing to find the causative agent, he got disinterested in the work and eventually abandoned it. Bajering took over from where Ivanovsky left. He decided to do the experiments more systematically. Results of Ivanovsky's work had ruled out bacterial cause, at least any known ones. Bajering decided to study infection characteristics of filtrate much more thoroughly. For this, he worked with dilutions of the filtrate. This video presents simplified reenactment of his experiments. Tubes in the video are filled with 9 ml of sterile diluent. Now one can take 1 ml of filtrate and add it in tube A, making a total of 10 ml. So it is a 1 in 10th dilution. Then 1 ml is shifted from tube A, which is already diluted 10 times, to tube B. In the third step, 1 ml is taken from tube B and transferred to tube C. And finally, 1 ml is moved from tube C to tube D. When 1 ml from flask is transferred to tube A, final volume becomes 10 ml. Therefore, dilution becomes 1/10. 1 ml from tube A, which is already diluted 10 times, is further diluted 10 times upon insertion in tube B. So, sample in tube B is 100 times more diluted than the filtrate in flask. In the same manner, dilutions in tube C and tube D become 1000 times and 10,000 times more diluted than the filtrate of flask. From tube A through tube D, concentration of infection-causing agent is reduced 10 times at every step. Filtrate dilutions are inoculated on plants, and then one has to wait until symptoms of tobacco mosaic disease become apparent. On day 2, plant which was inoculated with most concentrated filtrate shows the signs of disease. Then on day 4, symptoms appear on the plant that was inoculated with tube B. On day 7, plant which was applied with tube C showed the disease. And finally, even the most diluted sample from tube D was able to cause disease on 10th day of inoculation. We can see that with decreasing concentration, appearance of symptoms gets delayed. But once disease is there, it comes with same intensity in all 4 cases. How can you explain these findings? Ivanovsky's work excluded the possibility of bacterium. We are left with 3 other assumptions about the nature of disease-causing agent. Might it be some toxin secreted by bacterium? No, this could not be possible because if a bacterial toxin could cause disease, why not bacterium itself? We know that bacteria retained by filtration could not cause tobacco mosaic disease. Second assumption of it being some other toxic substance is also improbable. If it was a poison or toxin, there must have been a dilution where it would have become ineffective. Therefore, observable symptoms of disease should not have appeared in plants inoculated with most diluted samples. What he observed instead was that even the plants which were sprayed with possibly most diluted filtrate got the disease. If it was not a bacterial secreted or any other toxin, what was it? Only possibility was that it could be some yet unknown, bizarre living thing, as plants got the disease even when inoculated with least concentrated filtrate. Delayed onset of disease in these plants can only make sense if disease-causing agent is living, which means it increases in number as the infection proceeds after inoculation. To explain it further, let's compare how infection should proceed when plants are inoculated with most concentrated versus least concentrated filtrate. In this thought experiment, we spray one leaf with filtrate from tube A and other with filtrate from tube D. As we apply the filtrate on 2 leaves, filtrate from tube A occupies most of the cells in the leaf, but diluted filtrate from tube D can only invade few cells. Causative agent then starts the infection cycle of the invaded cells, meanwhile damaging them, and multiplies at the end of infection cycle. Agent then leaves the damaged cells and invades neighboring healthy cells, also damaging them at the end of life cycle. In this case, infectious dose was high, which meant that the significant portion of leaf got damaged in just 2 infection cycles. In the other case, however, very small portion of the leaf is damaged, which is still invisible on the leaf. Although on both leaves 2 infection cycles are completed, but leaf with greater initial infectious dose shows signs much earlier. In 10 days' time, plant infected with extremely diluted dose also shows visible symptoms of tobacco mosaic disease. Pagering conducted several other experiments to prove non-bacterial nature of tobacco mosaic disease. Here I would mention 3 more. He heated filtrate but found no significant loss of infection-causing ability after heat treatment, something highly unlikely in the case of bacteria. He then added ethanol in filtrate to test survivability of infectious agent in ethanol. Again, he couldn't observe significant loss of infectivity, which is highly unlike bacteria, which can be killed easily in ethanol. Finally, he transferred filtrate onto an agar column. Agar is known to retain bacteria on surface, but fluids can pass through agar matrix. He then took agar plugs from lower layers and showed that those could cause disease in tobacco plants just like the filtrate, while material from the agar surface did not cause tobacco mosaic disease. Because diffusion into agar was considered property of fluids and soluble substances, he concluded that filtrate contained a fluid which was a living object. He termed it contagium vivum fluidum, which literally meant an infectious living fluid. To distinguish it from bacteria, he named infection-causing agent virus. So Bajering is credited for discovering new form of infectious agents. But it was W.M. Stanley who worked out true nature of viruses. He obtained the filtrate and crystallized it using known methods for the proteins. Through X-ray crystallography and electron microscopy, he conclusively proved that virus was not a fluid object, but discrete entity chiefly made of proteins arranged in specific structures. Today we know that tobacco mosaic virus is composed of helical capsid which results from multimerization of proteins called capsomeres. Beneath the capsid lies a plus-stranded RNA molecule that is store of structural and functional information for the virus. This is the first of the 3 videos on the discovery of viruses. Upcoming 2 videos will narrate visual tales of discovery of first bacterial and first animal virus. I'm sure you are interested to know these stories. You just need to subscribe to my channel and hit the bell icon to get notified when new videos are uploaded.